Well, thank you very much to Caetano and to Eloisa for the invitation. So as I said, my name's Mike Templeton. I think I was probably invited to this talk because I'm Canadian originally. So I was born in New Brunswick, grew up in Newfoundland, went to the University of Toronto for my university education, and then after my PhD, I moved over to the UK. Where I'm currently Professor of Public Health Engineering in the Department of Civil and Environmental Engineering at Imperial College London. I also have a research chair currently with Oxfam and Water for People, who are two not-for-profit organizations you might have heard of before, and that's focusing on various aspects of sanitation technology. But actually, the topic I'm going to talk about today is not so much related to that chair. It's more related to some work I've been doing for the past 10 or 15 years, looking at the role of WASH for combating worm-based diseases, parasitic worms. So you can see my talk, Deploying WASH in the War on Worms. I should say I don't have a bias. I don't hate all worms. In fact, I'm very keen on some worms. I've done a lot of work on worm composting toilets, for example, in sanitation, where the worms can actually transform fecal matter into compost in situ and make it much easier to empty toilets and much safer. So some worms are great, composting worms, but here we're focusing more on the ones that that cause a range of illnesses in humans, so the parasitic worms. So there's my email address. If you don't get a chance to ask a question, feel free to email me anytime. Okay. So why talk about worms? I think, first of all, because I think it's a really understudied topic in environmental engineering. And certainly when I went through environmental engineering as a undergraduate and then a graduate student, we always focus on bacteria, viruses, and protozoa when we talk about water supply. And I didn't really hear very much about worms when I was in university. However, if you think about it, some of the most common debilitating diseases in the world are actually caused by parasitic worms. Some of which are called schistosomes, some are called helminths, and a number of these are what are classed by the WHO as neglected tropical diseases, or NTDs. And you could say that neglected tropical diseases are actually more about neglected people than diseases in some ways, because a lot of them affect the poorest people in the world, the most neglected people in the world, let's say. They're maybe not as sexy as diseases like malaria and HIV/AIDS, which have had a lot more focus in terms of elimination efforts. But if you look at the numbers of people around the world affected by these parasitic worms, it's really staggering. So it's a huge problem out there that a lot of people don't know about, or they don't talk about, or they don't fund as an activity. And there's a huge role for WASH. WASH has a crucial role really to play to protect people from parasitic worms. So I really, my plug, I guess, if you don't take anything else from the presentation, is that I think WASH engineers need to get much more involved in this topic. And to specify in this presentation, I'm mainly going to be focusing on schistosomiasis and soil transmitted illness. So in the war on worms, here are the enemies that we're going to be mainly talking about today. And this is only a few of the worms that are out there. But I'm mainly going to be talking about schistosoma, but also I'll be saying a bit about STHs during the presentation. So those are things like Ascaris and Trichuris. And they tend to have one form which infects you or exists in the environment. So in the case of schistosomiasis, it's these things called stercari, which are microscopic things which swim around in the water and penetrate your skin if you're in contact with the water. And then once that form of the worm is in your body, it then develops into an adult worm, and that's where a lot of the problems start to happen in your body. So I'm actually giving a keynote talk in Brazil on Monday at a symposium on schistosomiasis, because it's still a disease that I think infects about 6 million people in Brazil. And so you're getting a bit of a sneak preview today of what I'll be saying on that on Monday. And I'll say a bit about the other helminths at various points in the talk also, but mainly I'll focus on schistosomiasis. So I don't know if you've heard of schistosomiasis before. So another name is Bilharzia, named after the scientist who sort of discovered the disease and the life cycle. So it's different from other diseases which you acquire through ingestion. It's when you're infected with one of these worms, you shed the eggs either in your urine or your stool, and quite often that causes a lot of bleeding as the eggs leave your body. Those then go into water bodies if you don't have proper wash, and you can have intermediate hosts getting infected. And the intermediate hosts then multiply the vector of the disease and lead to other people becoming infected when they come in contact with water that contains those cercariae that I showed on the previous slide. So again, you might not have heard of this disease before, but over 200 million people have it in the world, at least, and it's estimated that maybe about 200,000 die from it each year, and about 90+% are in Africa, but there are also other pockets around the world. And it causes a range of symptoms, as I said, bloody urine and diarrhea, abdominal pain, enlargement of the liver, inflammation, scarring of the bladder. So it causes a lot of internal damage. And in children, it's also really been shown to have a lot of negative knock-on effects on learning, but also in terms of stunting growth and not being able to take in nutrients. And so really, especially a lot of the efforts on schistosomiasis control are really focused on trying to prevent kids from getting it at an early age. And the good news, I guess the good news about schistosomiasis is there is a treatment for it. There's actually a few drugs which can be administered and are administered on huge scales. And to the pharmaceutical companies' credit, they actually do provide these drugs at very low cost. I mean, there's still a cost to distributing the drugs, but they're very effective. The problem with these drugs is that they're not a vaccine. They're deworming drugs, and if you're re-exposed to the parasite, you're going to become reinfected again. So that's where WASH really has an important role to play in this disease. It is really stopping that cycle of infection and reinfection alongside drug administration. And why is this a particularly timely topic is that right now the WHO has sort of said, we're really sick of these neglected tropical diseases. We want to move towards talking about elimination of a lot of them. So not just controlling them, keeping the numbers the same or reducing them slowly, but actually trying to eliminate them. And to do that, we have to sort of move away from the historic approach, which was really just trying to get rid of it through drug administration and really looking at it in a more integrated cross-sector approach, including WASH as part of that. So again, apologies if you already know a lot about schistosomiasis, but there's a few different species, and the main ones are Mansoni, Hematobium, and Japonicum. And you can see their distribution around the world, but mostly in Africa. And you can see, especially down the Nile River Valley there, quite a lot of schistosomiasis there. And historically, people have even gone back and found that the ancient Egyptians suffered from schistosomiasis, and when Napoleon's army invaded Egypt, a lot of the French soldiers got schistosomiasis. Some people think Napoleon himself actually might have gotten schistosomiasis. So it's what people in international development call a wicked problem, because it's been around for centuries, if not millennia, really. And just going back to thinking about the life cycle. So this is from the Centers for Disease Control representation of the life cycle. So again, you can see the person in the middle and shedding the eggs which hatch, releasing Mericidia at number 2 there, and then it's going into the snail, a particular type of snail, and then those snails shedding these things called cercariae into the water, which are what penetrate into skin of other people. And the really important thing to remember is that it only takes one of these eggs getting into the water to infect a snail. And over the lifetime of a snail will probably shed about 20,000 cercariae if it's infected. So that's a really important and maybe underappreciated aspect of the life cycle, is that the snail plays a huge part in magnifying the level of risk. And really, if you have even just a small number of eggs still reaching the water, you're going to have a high risk being maintained to humans. So it's definitely not a one-to-one relationship. And you really have to achieve high levels of protection of the water body to get a meaningful reduction in the risk to humans. So this is why it's such a tricky thing to eliminate. So what do we mean when we say wash for schistosomiasis? Because there's actually a lot of stuff online and from WHO and other organizations saying, of course, you need to have wash to prevent schistosomiasis. But I think there's a lot of detail that needs clarification about that. So what type of wash, for example, or what wash characteristics or conditions are needed to be preventive against schistosomiasis? And because of the life cycle, the wash that you would choose if you were looking to prevent schistosomiasis would be quite different probably than the wash that you'd need for preventing fecal oral diseases. So it's, from an environmental engineer standpoint, that was one thing that really interested me initially when I got into this. I'm not going to say too much about the S in wash in this presentation, the sanitation bit, for two reasons. One is that I think as long as you have a toilet that is able to isolate the waste, so a form of improved sanitation, then that waste should not reach water bodies, and therefore you're doing an excellent job of cutting transmission. So from a research standpoint, I don't think, personally, I don't think there's a lot of interest or need out there to design better toilets from a schistosomiasis standpoint, as long as you can achieve an improved toilet in terms of how that's already defined. But the other reason I don't say too much about sanitation is that I think in many cases, it's only going to really be an effective intervention in the long term when you have almost universal coverage and usage of sanitation. And going back to the context of the life cycle, that really makes sense. So like I've seen studies where people have said, well, we went in and we gave 50% of the community toilets, and we measured no impact on schistosomiasis. And that's totally what I would expect, because if the other 50% don't have sanitation, because of that effect of multiplying the risk of the snail in the life cycle, I wouldn't expect the reduction in risk of schistosomiasis to be very significant. So I'm not saying sanitation is not important. Obviously, it's hugely important for other reasons that have nothing to do with schistosomiasis. But in terms of this particular disease, it's probably going to be something only in the longer term when you achieve near universal coverage. So from that standpoint, a lot of the communities that we were involving in our research and talking with, that thing of universal sanitation coverage was not going to be happening anytime in the near term. So the approach that quite often was needed was looking at just reducing contact with infested water in the short term, in the near term. So assuming that the water is infested, how are we going to achieve safe water for those communities? And so focusing on reducing contact with infested water, we need to provide not just safe drinking water, but water for all needs. So washing, recreation, and that will be quite variable in different settings. Reducing water contact doesn't mean simply providing clean new water supplies also. So for example, lots of communities where boreholes have been dug to provide water that's not contaminated by the schistosome cercariae, but then you have fishermen or you have people swimming in the river who have clean tap water but are still at risk. For a long time, mollusciciding was seen as another important intervention for schistosomiasis, so try to kill all the intermediate hosts. But in some ways, this is a lot like the sanitation argument, that unless you get nearly 100% elimination of snail hosts, the risk is going to remain high in terms of the contact with contaminated water. And also, there's been a lot of concern about the ecological impacts of mollusciciding, of just wiping out one species from the ecosystem, and what are the impacts on other forms of aquatic life. And then finally, even if we do kind of get the whole reducing water contact reducing contact with infested water, if we solve that problem, nothing really works without communication and public understanding of the risk. So lots of cases where NGOs have gone in and said, this water is safe, that water is not safe, and people continue to use the unsafe water for a range of reasons that would have been obvious if they had just spoken to the community and understood why they go to that water source. So for example, there's a case in Tanzania where the women would gather every morning to wash their clothes together at the lakeside because it was part of their social routine. And they didn't want to go to, sort of into the community to use the borehole that had been dug because everybody couldn't really gather around that at the same time. So I'm an engineer, but I think the whole behavior and communication side of public health is as important, or if not more important, than the technical side of it. So I'm going to give you a bit of summary of sort of my group's work on WASH and schisto over the past 10 years or so, and it really started with 10 or 15 years. It really started with some systematic reviews which were published, which looked at all of the evidence that existed out there about the impact of WASH on schistosomiasis. And alongside that, we were able to partner with a number of organizations who were looking at child health in Ethiopia, and they did a big national mapping program looking at the links between aspects of school facilities for WASH and STHs and schistosomiasis. So I don't know how many of you have heard of systematic reviews or meta-analysis before. If you're from a medical background, you probably have heard of this because it's a very common research technique, but it's basically where you go through a systematic process of reviewing literature using keywords, and you have a systematic way of filtering out the papers that you find through that search to limit it to ones that have certain characteristics. And then you're able to actually combine the data from those studies and come up with some pretty powerful conclusions by using a much larger data set by combining those studies together. So we decided to do that for WASH and schistosomiasis. So we looked at all the studies that have been published up until about 2013 or 14. And there were some that looked at only kids, some that looked at adults and kids, some that only looked at adults. There were some that looked at only hematobium, some at japonicum, some at Mancini, different countries, but we then pooled all of that data together and calculated odds ratios in terms of, does water, sanitation, hygiene have a positive or negative effect? So it was about 44 studies, about 90 data sets. And what this statistical analysis found was that increasing access to safe water and sanitation can indeed be important measures to reduce the odds of schistosome infection, which sounds good. It's what we expected from the life cycle. And I think whenever you do a systematic review, it's, as I say, it's more about providing the evidence based on all of the knowledge that exists currently and making a really strong case for something that is really important from a public health messaging standpoint. However, we did find there were a lot of common limitations of previous studies and on the impact of WASH and schistosomiasis. One was that there's a lot of socioeconomic confounding. So a lot of studies were looking at very wealthy populations and very poor populations. And where water contact, for example, or sanitation was not the only important difference between the populations that were included in the study, or where a lot of the people from the poorer populations had a lot more occupational exposure, for example, to water. People who are irrigation farmers or fishermen being compared against bankers or doctors who hardly ever had to go into the water. So there was a lot of socioeconomic confounding in a lot of the studies. And also, I don't know if it was because a lot of the studies were done by medics, but there was really a poor description of WASH in a lot of the studies. So it was kind of like, yeah, those guys had toilets, or yes, they had tap water, but nothing about the status of them. Were they clean? Were people actually using them? Had they broken down? So that there wasn't a very complete and useful explanation of the status of the WASH in a lot of the studies, which we thought was also a bit of a confounding factor. So as I said, as those reviews were going on, we were able to be part of this large Ethiopian national mapping study, and we tried to address some of those confounding factors in this large study. And it included about 80,000 kids, so it was 50 kids in about 1,600 schools across Ethiopia. And it was in partnership with the Ethiopian Public Health Institute and the Partnership for Child Development and SCI, the Schistosomiasis Control Initiative. And the partners were collecting a lot of health information on the kids, and our role was really to devise and apply some WASH scores, so to be a lot more descriptive of the quality of the WASH in the schools. And then once you have sort of a numerical scale for quantifying the quality of the WASH, then you can do a lot of statistical comparisons with a lot of the health metrics that were being measured, which you can't do if you just take a very high-level qualitative approach to to describing the status of the wash. So what was the result of the study? Well, before I get to that, so what were these scores, these metrics that we developed? So there's a water score, which included things like frequency of water collection by students. A lot of these schools were by rivers and didn't have other sources of water supply, so the kids would be sent to get water from a river or a lake several times a day to bring water back to the school. It also included asking about the source of water in the wet and the dry season. So sometimes these schools would have access to rainwater, for example, as a source of water in some seasons, but not in others. So the exposure would vary over the course of the year. Sanitation score included things like whether the latrine blocks are gender segregated, what type of toilet they had, were there doors on the toilets, how clean were the floors and walls, presence of odors and flies. So not just are there toilets and do boys and girls have equal access to toilets, but also things about the quality of the toilets. And then the hygiene score included things like, do people have water available for hand washing? And is there actually soap available in the schools? So these things all fed into a score. So each of these questions, so each school was surveyed in a systematic way where they were looking for assigning a score to each of these different questions, which then you could sum up into an overall sanitation score for a particular school. And so the findings were really important, I think, in that it showed, first of all, we found that for Mansonii, schistosoma mansonii, the strongest association was with water contact and no significant association with sanitation. And, again, you might think that's kind of a frustrating message to say that, to imply that sanitation is not important. And that's not what I'm saying. But what we're saying is that, again, going back to the life cycle, it probably makes sense, actually, that sanitation, you wouldn't expect to see a huge impact unless there was nearly universal coverage and usage of sanitation within a community. So probably just looking at schools was not enough, maybe, to say that sanitation was important in that case. But interestingly, there was a statistical link with the amount of time that the kids had to go and collect water every day, which is what we expected. And interestingly, also, the study included a lot of other soil-transmitted helminths, and there were links to the quality of the sanitation, as stronger links than with schistosomiasis. And again, that kind of makes sense because a lot of these helminths are spread by dirty toilets, which can actually serve as a focal point for disease. If kids are going to, a latrine block where there's a lot of fecal matter on the ground and a lot of eggs of the helminths on the ground, and that's actually a focal point for where kids are getting infected. So I think from those studies and from the systematic review, really going forward, we then decided to take more of a focus on reducing water contact, as I said. So assuming that the exposure to water is something that people need as part of their daily lives, and that water is quite often going to be a high risk based on the wash situation in the broader community. So how do we reduce risk to contamination, contact with contaminated water? So the first option, and I think this is something that even if you're not thinking specifically about schistosomiasis, you would probably say is the first option anyways, is to look for protected groundwater or rainwater. So don't think about having to treat the water, but look for sources of water that are unlikely to be already contaminated. And the slight difference here with schistosomiasis is that it has to be a water source that can provide all of the water needs for the community. So not just drinking, but also laundry, bathing, etc. And then barring that, if you can't find that, then the option 2 would be, well, you have to provide some way of abstracting water from surface water and treating it to provide a safe alternative water source. And no matter which option you consider, I think that has to be accompanied by interventions which also think about reducing occupational exposure, as I said earlier. So maybe that's about providing boots and gloves for fishermen. Or there was actually a really neat study in Ghana several years ago where they looked at the impact of putting in a chlorinated swimming pool in a school so that the kids could swim in the swimming pool instead of having to go in the lake nearby. And that actually had a measurable, important reduction in infection. So these other types of water infrastructure, are going to be really important besides the standard sort of taps and toilets that we think about in the WASH community. Awareness campaigns, of course, about the risk of exposure to contaminated water, and then plans to uptake and maintain the new infrastructure and involving the communities and all the decisions that go along with in any new infrastructure. So also in that process, we identified a few gaps in knowledge in terms of if we were to try to provide water infrastructure for these communities, what were some things we didn't know? And from an environmental engineer's standpoint, there were some things which are sort of right in our wheelhouse, or what are things that we're used to looking at as environmental engineers. So like, I couldn't even tell you at the time what would be the chlorine dose you'd need to add to water to make it safe, or how would you design a sand filter, or can you just store the water for a certain amount of time, and will that reduce the risk? And so at the time, at the end of this period, we lacked a lot of information that would be needed to really provide the detailed, robust design advice. And then also we were interested in, can we develop better ways of detecting the cercari in the water bodies? So there isn't really a great way of assigning a risk level to different water bodies based on any kind of sampling or scientific evidence. And we worked with some synthetic biologists to develop some new bioassays to sample water for the parasite. I won't say too much about that today, but I think that's still a research need that's out there. And then there was a non-technical part about how do we sustainably implement these solutions in communities, and what are the behavioral or knowledge barriers to implementing these types of infrastructure. And then finally, how do we get the WASH and MDA, the mass drug administration sectors, working together on this? Because in a lot of countries where schistosomiasis is endemic, we found that there's quite often a Ministry of Health and a Ministry of Water, which are not working or not talking to each other. And it just makes much more sense if you're going to try to tackle one of these diseases for more integrated approaches. So we tried to investigate these questions through a project that was unfunded in 2017 called the WISER project. And the website is still active if you want to check it out. So WISER stood for Water Infrastructure for Schistosomiasis Endemic Regions. Whenever you're going for big funding, you always have to think of a catchy acronym that the reviewers will remember in their minds. So we went with WISER. And it was about a £1.4 million funded project, and it had partners in the UK, Ethiopia, and Tanzania. And it was a really fun project to be part of because it was very interdisciplinary. So it had engineers, but also parasitologists, snail experts, anthropologists. There were even actors involved. So I'll explain why actors were involved a bit later on. But it was really trying to answer a lot of those knowledge gaps I just mentioned, and also really had a lot of stakeholder workshops and community engagement in Ethiopia and Tanzania. I thought another title for the project could have been Adventures in Snail Hunting, because the researchers did spend a lot of time out, especially from the Natural History Museum, who are experts in this, looking for the intermediate host in different types of water bodies. One of the things I really learned and developed a lot of respect for my colleagues from the Natural History Museum is you really have to know what you're looking for in terms of what type of snail is the host. And this is going back to why assessing the risk level of a particular water body is not straightforward, because first of all, you have to find the snail that is the intermediate host, and then you have to find out how many of those snails are actually infected. And that's requires a huge amount of expertise and a huge amount of time and effort to do that. But what we did in our lab at Imperial was we basically used the aquatic snails as sort of like a supplier of the parasite. So we had infected snails in the lab, which were shedding the cercari at high rates. So we were keeping these snails alive, sick, but not dead, because we wanted them to be shedding the cercari. And then we could use the cercari for answering a lot of those water questions that I posed earlier on, those water treatment questions. So again, when we started the water treatment research, we started with a systematic review. So we did another review this time on the papers that looked at the links between different types of water treatment and schistosome cercari. And we found about 67 studies over 116 years. And they studied a range of different treatment methods and conditions, as you can see listed there. But really, again, our conclusion overall was there's been a lot of academic stuff done. There's been a lot of stuff done in the field under less robust conditions, let's say, but really still not enough information to be able to tell someone, here's how you design a filter, for example, or here's how much chlorine you need to be safe, to be sure that the water is safe from schistosome cercari. So we did a series then of experiments on different types of treatment methods. So with chlorine, we looked at how much chlorine is required, what concentrations and contact times, and what's the effective pH and temperature. So all the kinds of things environmental engineers would look at for other pathogens in water. Again, the idea here is that if somebody is supplying water, for example, in jerry cans, or if they're taking water out of a contaminated water body and then bringing it back home, chlorine could be an intervention at that stage. We're not talking about dosing chlorine into Lake Victoria or something like that and causing all kinds of ecological issues. It's about taking water out of the environment and then treating it with chlorine at the point of use. So a bit of information about the experiments we did. So on 50 ml water samples, we put about 100 cercari in each one, range of temperatures and pHs. We used bleach, which is the more common form of chlorine in a lot of endemic countries, and we looked at sort of contact times that would be reasonable in real life. And so if a lot of you are environmental engineers, you probably know the CT value, which is used in chlorination. So CT is the product of chlorine and chlorine residual and contact time. And that's sort of the value that's used to quantify how much disinfection you've provided to water. So for example, if you have 1 milligram per liter of chlorine residual for 30 minutes, the CT value would be 30 times 1. And so a shot here from one of the papers that was published, and we showed that the CT values, so a higher CT gives you a higher log inactivation of the cercari, as you'd expect. And we did this in the lab, but also using water taken from Lake Victoria with our colleagues in Tanzania, and found that the actual cercari in the actual water of Lake Victoria, the CT values that were required were pretty similar to the ones that we had in the lab under similar pH conditions. And so there's Laura Braun was the PhD student who published a paper on this, and I've just cut out one of the tables there from the paper to show the CT value for Schistosoma mannsoni cercari from that study. And basically, it's a fairly easy organism to kill with chlorine, not completely out of the range of other waterborne pathogens, slightly harder than bacteria, but a CT value of 30 would give you about 2 log 99% inactivation. And we actually had people when this work was going on who would email us occasionally, people who are traveling through Africa, for example, and saying, we're driving through Africa, we have a Jeep with a big water tank on the back that we keep filling up at lakes. How much chlorine should we add to make it safe so that we can use it for washing our hands and taking showers occasionally? And as I say, before this study, I didn't have a clear answer for them, but it seems like 30 milligrams minutes per liter should give you about 2 log inactivation. So that was one of the main recommendations from that work. The ongoing challenge is that there's no simple way of measuring chlorine residual in a lot of really low income settings. And so our recommendation was really that to achieve that required CT value, maybe ministries of water need to provide guidance which is based on the local form of water supply in those communities. So, for example, in a lot of places in Ethiopia and Tanzania, we noticed that the main source of a lot of household water was from water vendors who were collecting it from a particular water source and then distributing it in sort of jerry cans. And if that was something that was systematic and regular, then you probably could come up with some easy recommendations if you could ensure a chlorine supply of bleach to the water vendors. We also looked at sand filtration experiments. So again, how deep does the sand need to be? What size of sand? How quickly can you pass the water through the sand to make sure you're removing all the cercari? And again, paper published on that. And yeah, I guess again the take-home message is it can be done using the types of sand that you can find typically in these regions. But some specific advice about the filtration rate and the minimum depth that you need to get good 100% removal of cercari. But filtration works. It's an effective way to protect from the cercari. We also looked at UV, and initially we did another systematic review. So this was looking at UV not just with schistosomiasis, but with a range of different worms, parasitic worms. So also looking at Ascaris and other types. and looking at SODIS, so solar disinfection, but also other wavelengths of UV light. And again, a wide range of data out there, a wide range of methods, a wide range of methods in terms of quantifying the dose that the organisms were receiving, and also in terms of measuring whether they were dead or whether they were sort of damaged but not infective anymore. So a bit of a complicated picture. My student Lucinda then did a lot of experimental work looking at different wavelengths of UV light and determining the effectiveness and the dose that you need for either 1 or 2 log inactivation at different wavelengths. And sort of the take-home message from this is that you need pretty high doses actually of UV compared to other waterborne pathogens to get the kill of the organism of the cercari. But actually, she found that if you expose them to UV, but then you wait a certain amount of time, that actually the inactivation rate, like after 180 minutes after exposure, was actually quite a bit higher. So I think we came away from that study sort of saying UV is probably not going to be a main source of protection against schistosomiasis infection, because you do need quite high doses. But it's clear that the UV is damaging the parasite, and they do die after a certain amount of time post-exposure. And then also we've done a lot of work on soap. So looking at the impact of soap use, because again, there's a lot of sort of anecdotal evidence out there that soap is protective against schistosomiasis. And in Ethiopia, there's been a lot of traditional use of a plant called endod, which is a type of soap, is used as a soap in Ethiopia. And some again evidence that it's protective against schistosomiasis. And so again, we did a systematic review looking at all the studies, which again suggested that soap does have a protective effect, but again, quite variable in terms of the types of methods that people have used, and also the types of soap that people used. But in general, the effect on cercari tended to follow a CT kind of relationship, the same as chlorine. So either higher soap dosage or longer contact with soap led to higher kill of the parasite. And then Jowdy, PhD student Jowdy Zhang, then did some more robust controlled experiments using powdered and barred soaps from Tanzania. And she determined, again, the lethal concentrations and times that you need to make the water safe from cercari. And the take-home message here was that actually soap is toxic to the cercari, but it's not 100% effective under the types of doses that you would typically be using. It's maybe sort of in the 50 to 60% kill range at the typical doses that you'd be using most of the time. So soap is protective, but I wouldn't bet my health on being fully protected by using soap. And just a few more slides, I guess one about the limitations of wash on its own. So, even if we know exactly how to design the most effective types of wash, these will be ineffective without community engagement buy-in, as I said earlier. And so if people have access to safe water, but they still prefer to use contaminated water for social or cultural reasons, then the safe water access is ineffective. We saw this in a lot of communities. And so the final part of the WISER project was actually working with a charity called Acting for Health. So this is where the actors came in. And they have this really neat approach that they use where they work with communities to develop effective public health messaging for their own communities using theater-based education campaign methods. So there was a paper we published on some work that we did on this in Ethiopia and Tanzania with Acting for Health, and it was really it was really an amazing experience because again, there's sort of this phase where you go into communities and the communities choose who are going to be part of this theater, and they actually develop the message. Typically, a fairly short message, but it's something that really gets to the heart of why the infection is such a problem in their community. And quite often these little theater vignettes, they came up with things which you would never know if you just did a traditional survey. So, for example, we worked in one community where they decided to talk about the fact that by taking praziquantel, the drug, there was this belief in the community that it caused impotence in men. And so they felt that really the big sort of thing that was driving infection was that people were not taking the drugs because they were afraid that it was going to have this effect on the men in the community. And so they developed a whole public health messaging play based on that, which was then shown to the whole community. It was also recorded, so part of the research was to also show whether showing films as opposed to live theater would be equally effective. But anyways, it was a really neat part of the project, and I think it really highlighted the fact that we do need some sort of creative ways to improve understanding the disease and also motivate communities to implement preventive measures. And finally, also to mention a study that was published in Nature Water recently, which was about the effect of unsanitary school latrines. So it was looking at the fact that there are a lot of schools around Africa, for example, that do have sanitation. They do have latrine blocks, but there's no process in place to keep them clean. And as a result, there are all kinds of health impacts that can be directly linked back to those unclean latrines. And so part of that study included a review of a range of different health impacts, including soil-transmitted helminths and schistosoma infections, but also other things. And it really led to a call for improved methods for what we would call school-led total sanitation. So there's a technique called community-led total sanitation, which is about generating motivation for communities to take control of the sanitation solution and situation and come up with their own solutions. And so this is about doing this at a school level and looking at ways that can involve kids in ways that are dignified and safe, but also are leading to sort of a health education at the same time. And it was a perspective article, but it was really sort of a review, and it also included a lot of nice examples of where effective school-based sanitation clubs and things like that have been good at maintaining latrine cleanliness, and hence had an impact on some of these diseases. And then finally, I think linking the WASH sector with the schisto community. So how do we do that? And I think this is still the thing that is limiting elimination efforts in a lot of places, is that the people who are doing the drug administration and the people implementing WASH are not on the same wavelength or know what is going on in the same place. So I guess the first thing I'd say to people in the schisto communities or the MDA community is find out what WASH organizations are already working in their regions, explain what schisto is to the WASH community, because I think it's probably low on the priority list for a lot of WASH implementers. And then, explain that WASH for schisto is not the same as WASH for fecal oral diseases. And in a few places, they've tried to do some mapping. So to try to look at where the disease is, where water contact is a particular risk, and basically look at where WASH is probably going to have the highest benefit to cost ratio in terms of tackling that disease. And then, quite often there's probably going to be like an additional component to WASH funding or additional sourcing of funds needed for some of the infrastructure that specifically is effective for targeting schisto. So, for example, it could be laundry basins, or it could be safe bathing or showering facilities. So the types of infrastructure that are not normally included in WASH funding, which tends to be about toilets and taps and hand washing stations, but some of these other water contact facilities that are also really important. And then finally, just 3 emerging topics in water and schistosomiasis. So one is that there's been a sad trend in parts of Africa recently where there's been a lot of hydroelectric dams which have been constructed, which are fantastic from a GDP standpoint and from providing energy security, etc. But it does also increase the risk of schistosomiasis by creating new ecosystems. And if you don't provide WASH to the communities alongside these hydroelectric dams, it actually, the dams themselves can become a focal point for infection if people are using that for water contact. I think there's still a need for better, easier methods for quantifying the risk of schistosomiasis in the environment. So similar to like quantitative microbial risk assessment that we would do in drinking water treatment, I think we need something equivalent for schistosomiasis. And there's also been a lot of interesting research recently on zoonotic transmission and hybridization between human forms of schistosomiasis and also, for example, bovine forms, which comes from cattle. But also schistosomiasis is known to infect other types of organisms like rodents. And if you're looking at eliminating schistosomiasis and you let's say you've achieved good levels of WASH in a community and you've achieved good levels of drug access, it could be the case that actually in some communities, the zoonotic transmission is contributing to the disease continuing to some degree. So I think these are emerging topics that people need to work a bit more on. So my take-home message is WASH engineers have a very important role to play in the war on worms within integrated strategies for neglected tropical diseases, such as schistosomiasis, alongside drug administration, health education, behavior change. I think you need to consider the life cycle of your target worm when selecting the most relevant WASH types. Just to summarize some of the scientific stuff I presented there. So Schistosoma cercari, they seem fairly sensitive to chlorine. They're pretty easy to remove by filtration. UV, not as effective. Soap is beneficial, but doesn't provide complete protection under the conditions that people would normally use it. And finally, that just to emphasize the point again, the WASH has to be adopted and maintained over time to be effective, which is not something specific to worms, I think. But I think something that is specific to worms is that really community level coverage is something that's really crucial. So I think, sometimes when people are focusing on fecal oral diseases, they look at individual access to WASH. But if you're as I said earlier, when describing the transmission cycle, if you're talking about schistosomiasis, for example, it doesn't only matter if you have access to WASH, it also matters if your neighbor has access to WASH in terms of the level of risk to you. Okay, so that's, hope that was interesting and motivated some people to get into this topic, and happy to answer some questions.